Hi Year 12s. This will be the last video that we look at in the DNA and proteins topic. This is the second video for our PowerPoint on genetic engineering, and in this PowerPoint we're going to be focusing on the process of CRISPR. So what exactly is CRISPR? Well, first off we need to know that CRISPR is actually an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it's a naturally occurring defense mechanism the bacteria use to fight off viruses. CRISPR provides bacteria with resistance to viruses called bacteriophages, which helps to recognize, and it works by recognizing and destroying viral DNA that it's encountered before. All that CRISPR actually is a length of DNA that's found naturally in the genomes of bacteria in a section that we call the CRISPR array. There are nucleotides on either end of each half of the sequence, so these parts in black here, which are complementary to each other. So C will go with G, C will go with G, G will go with C, U will go with A, etc. Because they've got these complementary ends on both ends of the structure, or the DNA strand, we can fold it up to create a hairpin-like structure. So it forms into these, which is a hairpin-like structure. Exactly the same one, just being folded into that structure. The repeat sequences here, which are complementary to each other, are actually separated by something called a spacer, which is just a short length of DNA that matches DNA in viruses and therefore allows it to attack the bacteria. Viral DNA will be cut and inserted into the bacterial genome by something called Cas proteins. What are Cas proteins? Cas proteins are just enzymes that break down nucleic acids such as DNA and RNA. Basically what they're used for is to cut and paste viral DNA into the CRISPR array to create a new spacer, as well as helping to break down invading viruses using the information from their contained spacer. How do we use CRISPR and Cas in biotechnology? So obviously this is a really good method for bacteria, but how is this useful to humans? Well, it's becoming a widely used tool in genetic engineering because of the ability to insert or remove any sequence of DNA from the genome of an organism. So we can use CRISPR and Cas enzymes to basically take any sequence of DNA we want and remove it from an organism. Most normally in humans, we use a system called Crispr/Cas9. Cas9 is exactly what it sounds like, CRISPR-associated protein 9, CAS9. Can cut any DNA sequence from the genome of an organism when it's paired with something called a synthetic guide RNA. Or that's another type of RNA we haven't talked about, gRNA, guide RNA. The gRNA molecule created will have a complementary base sequence to the target DNA sequence that we're looking for. Cas9 will then cut the sugar phosphate backbone of the DNA at those specific locations, and the genome of the host will be edited by inserting or removing DNA nucleotide sequences. So CRISPR-Cas9 is the process that we're using in humans. What are the benefits and limitations of CRISPR/Cas? So the benefits are that genomes can be edited more rapidly, a lot faster than the conventional techniques that we've come up with to date. Multiple genes can be edited at the same time. It's very cheap technology in terms of biological technologies. Gene editing is highly specific and accurate, so we're actually pinpointing the exact sequence we're looking for. And the guide RNA sequence can be altered to be complementary to any nucleotide sequence that we want to target. The limitations, unfortunately, are that the mutations in target nucleotide sequences can reduce the effectiveness of Cas9. Cuts made by Cas9 can be repaired before the editing can actually occur, and unwanted mutations can occur. Probably a good time to talk as well, CRISPR is very widely used in biotechnology. There is a new process that was looked at last year called Majestic. I'm not going to talk about this PowerPoint because it's not in your syllabus, but Majestic is very similar to CRISPR. Might be worth having a look. In fact, we might even do a task on Majestic this week. So, might be one for you even to consider when it comes to your Science as a Human Endeavour task later this year. Majestic. First off though, what are the applications of CRISPR? So we can use it in agriculture to insert genes which allow plants to become disease resistant. In biotechnology, we can modify yeast cells to synthesize hydrocarbons to help us make plastics. In terms of research, we can create mutations in certain genes to help us understand genetic disease. For gene therapy, we can mutate genes, allowing scientists to correct them. In immunology, allows us to remove HIV DNA from genomes of infected people, so we can actually try and physically remove HIV DNA. What are the ethical concerns with CRISPR? First off, what happens if there's an accidental or deliberate release and airborne viral vectors escape the lab? There's lots of possible concerns there. Off-target editing. CRISPR can edit the genomes of organisms at unwanted locations, creating mutations we don't want. And lastly, design of babies. So eventually will this be misused to create genetically superior humans, creating a super race? All things we have to consider when we're dealing with CRISPR and Cas.